shrimp are not closely related to the shrimp we eat. These popular, odd-looking creatures are advertised as an easy-to-care-for pet that grows quickly and does not have a very long lifespan. We will briefly discuss the life cycle, growth conditions, uses, and sample researches involving brine shrimps. Artemisia is the scientific name for brine shrimps, or commonly known as sea monkeys. Brine shrimp belong to the domain Eukaryota, kingdom Metazoa, phylum Arthropoda, subphylum Crustacea, and class Brachiopoda. Brine shrimp do not live in the oceans, they inhabit inland salt lakes. The cyst can lay dormant on the sides of the lake until the salinity reaches a desirable level for hatching. They can remain dominant for many years. The only way to remove this shell is by oxidation with hypochlorite solutions such as bleach. The dehydrated cyst is concave in appearance, while the rehydrated cyst is spherical in shape. Adding the cyst in salt water initiate hatching. Just after hatching, the larval brine shrimp called a nucleus is no bigger than the period at the end of a sentence. To look for an evidence of hatching, you will suddenly see that the tiny dark points in the water are making jerky little movements. The nucleus grows fairly rapidly when conditions are favorable, such as food, oxygen, and the right concentration of salt, molting its outer shell frequently. It takes 3 to 6 weeks for the shrimp to reach maturity, and brine shrimps are capable of reproducing 300 new nuclei every 4 days. As they grow and develop, they undergo various stages, and each stage is separated from the next by a molt. The nucleus will molt about 15 times before becoming an adult in 2 to 3 weeks. The adult lives for another 4 to 5 weeks. Artemisia is usually about 8 to 10 millimeters, but can reach up to 50 millimeters depending on its environment. It has an elongated body, divided into at least 20 segments, and attached to its trunk are approximately 10 sets of flat, leaf-like appendages called philopodia that beat in a regular rhythm. The adults can be pale white, pink, green, or transparent, and usually live for a few months. They have compound eyes set on stalks and reduced mouth parts. The adult brine shrimps have a mouth, an anus, and an immature digestive tract. Adult female has an egg sac at the base of the body, while an adult male is distinguished by graspers near the head. They will be an average size of about 8 to 10 millimeters. Adults can live up to 4 months if kept in optimum keeping conditions. If kept at low salinities, females can produce free-swimming nuclei in preference to dormant cysts. For optimum growth, the brine shrimp's habitat must have the following conditions. Optimum water temperature for a 24-hour complete hatch is 25 to 28 degrees Celsius. Lower temperatures will result in a longer hatching time and inefficient hatches. Do not exceed 30 degrees Celsius. When preparing your hatching solution, a 30 to 35 parts per thousand or PPT salt solution is ideal under most conditions. Preferably, do not use iodized salt. A pH between 8 and 9 is recommended. Illumination is necessary to trigger the hatching mechanism within the embryo during the first few hours of incubation. Maintaining a light source during the entire incubation period is recommended to obtain optimum hatch results, and as mentioned above, for temperature control. Because they are attracted to light, they will congregate near the light you have placed at the side of the tank. All the naupliae will be attracted to the light, while the debris and unhatched cysts fall to the bottom. Constant aeration is necessary to keep cysts in suspension and to provide sufficient oxygen levels for the cysts to hatch. A minimum of 4 parts per million dissolved oxygen during the incubation is recommended. Strong aeration should not damage or hurt the brine shrimps or naupliae. The following can kill your brine shrimp culture. Overpopulation leads to low oxygen levels. Low oxygen produces more hemoglobin, resulting to brighter red color of cyst. Overfeeding produces high levels of dissolved nutrients, resulting to poor water quality. The presence of bottom and floating debris hampers movement and therefore starvation. And of course, lack of food. Because the digestive system of a brine shrimp is not yet fully developed in the first 24 hours of its life, no feeding is needed yet during that time. Growing brine shrimp are actually easy to feed since they are not fussy when it comes to their food. Their food is not directly consumed, but instead forced by the action of the swimming legs, which have small bristles on them, which catch and force the food to an area near the mouth. The movement of the swimming legs plays a critical role in ingestion of the food source and is why tanks need to be free of debris. One can feed them almost any type of food, such as whey, yeast, commercial fry food, soybean powder, wheat flour, fish meal, and even egg yolk. What is important is that the food used in feeding them is easily digestible by the brine shrimps, but not easily dissolved in water, which is why powdered milk is not an option. If you continuously expose the brine shrimps to light, it will increase its swimming activity, utilizing more energy, and may weaken the shrimps. Both the eggs and adults are used as feed for coral, larval fish, and other crustacea because of their low cost and ease of use. Brine shrimp are also useful in toxicity tests and for education purposes because they reproduce quickly and their environment is easy to replicate. They are used to teach students the proper technique to observe live specimens and how to design experiments to determine behavior, means of obtaining food, and most optimal environment for reproduction and development. Brine shrimp have been previously utilized in various bioassay systems. Among these applications have been plant extract toxicity, detection of fungal toxins, detection of heavy metals, levels of cyanobacteria toxins, and levels of pesticides. Bioassay or biological assay is a controlled experiment for the quantitative estimation of a substance by measuring its effect in a living organism. For example, The amount of the plant hormone auxin can be estimated by observing its effect on the curvature of oat coleoptiles. The concentration of the hormone is proportional to the curvature of the coleoptile. The primary purposes of bioassays are to measure the pharmacological activity of new or chemically undefined substances, as well as to determine side effect profiles, including toxicity. Instead of Hatch-Snoepli, some researchers focus on the inhibition of hatching of the eggs as the measurable outcome. For this assay, the cyst, which was incubated in a medium with different concentrations of organic extracts, and the percentage of hatching inhibition was determined. Toxicity was measured by comparing the percentage of Hatch-Snoepli to a control. The most commonly used assay for brine shrimps is the brine shrimp lethality assay. The brine shrimp lethality assay is an important tool for the preliminary cytotoxicity assay of the plant extract and others based on the ability to kill a laboratory cultured larvae, the nuclei. The nuclei were exposed to different concentrations of plant extracts for 24 to 48 hours. Lethality refers to the number of organisms killed or died. The procedure determines LC50 or IC50 values in micrograms per milliliter of active compounds and extracts in the brine medium. LC50 refers to the concentration at which 50% of the sample organisms are dead. Now let us try to determine the IC50 in this graph. Look for the 50% of the measured activity and determine at which concentration it intersects with the line graphs. For the 15-minute incubation, it resulted to an IC50 of 0.96 micromolars, while for the 5-minute incubation, it resulted to a 7.83 micromolars IC50. In some cases, when LC50 or IC50 can't be determined, the values are just plotted in a line graph to see a particular pattern or trend. The results are usually compared to a control. After the brine shrimp have gone through their life cycle, they can be poured down the drain. Any live brine shrimp can be fed to freshwater or saltwater fish. Eggs should be kept dry in their glass container and stored for later hatching. I hope through this activity, the students will not only increase their fascination, but also their respect for living animals, both from the conservation and welfare points of view.